Hi everyone, this is the second part of the third lecture for the Biofuels course entitled Selected Biotechnology Advances to Improve Industrial Biofuel Production. And we're going to start off here talking about Cvp organism development. Just to remind you, consolidated bioprocessing is the biological conversion of pre-treated lignocellulosic material by one or more organisms into a desired product in a single unit operation without added enzymes. It's also referred to as direct microbial conversion because you have a microbe or a consortium of microbes that directly converts your pre-treated biomass into a product of interest. So the main challenges for Cvp is that we require high levels of enzyme production without compromising ethanol fermentation, or if ethanol is our main product that we're interested in. We require co-fermentation of hexose and pentose sugars, which, as I mentioned, is a problem because of catabolite repression. We require our organisms to be able to tolerate relatively harsh process and product environments. And because of this, no natural organism has been isolated that basically has all of these desired properties. And we want to develop these through genetic engineering. So there have been several lines of thought on this, and Cvp organism development generally fall into one of two categories. People are either engineering cellulose producers, so organisms that are already growing on cellulosic material, and trying to get those to produce more of a desired product, such as ethanol, or they are producing organisms with very favorable product forming attributes, like the yeast Saccharomyces cerevisiae, which is the best ethanologen in industry, and trying to get those organisms to actually produce the cellulases to break down our lignocellulosic material. So we'll discuss first developing the yeast Saccharomyces cerevisiae for Cvp. It's a good place to start because a cerevisiae is used in wine and bread making, and also in first generation ethanol production. And this means that there's a lot of robust industrial fermentation knowledge attached to this organism. People really know how to do fermentation with Saccharomyces cerevisiae, it's a good idea to probably start with this organism and try and develop it for this new process of second generation ethanol production. So what does cerevisiae require in order to become a Cvp organism? It is a very good ethnologen. It means it makes a lot of ethanol relatively quickly, and it is quite robust in fermentation. It can survive even in relatively high ethanol concentrations, but it does require engineering to consume all of the sugars in biomass. It's a glutton for glucose. It doesn't use xylose at all, natural strains of cerevisiae. We needed to express cellulases, especially cellulobiohydrolases at high level. Now, just to remind you again of those three types of enzymes that we refer to as cellulases. They are the endoglucanases, which cut the cellulose within the amorphous areas, the less tightly, densely packed parts of the cellulose chains, and they provide free chain ends for the most important enzyme in crystalline cellulose hydrolysis called endoglucanases or cellulobiohydrolases to attach to. It attaches to those free ends. They're processive enzymes. They peel off these glucose chains from the cellulose crystal, and they release cellobiose in the process. The cellobiose then is degraded by beta-glucosidase into glucose, which can be fermented by the yeast or other organisms. So we need expression of those three different types of cellulose in the Saccharomyces cerevisiae. We need to produce these enzymes and consume the sugars in relatively toxic processing conditions. So why is it toxic? As I said, all of the pre-treatment methods that we apply in order to pre-treat our lignocellulosic biomass actually release some level of inhibitor into the fermentation. So our fermentation broth becomes or can become pretty nasty, and we're expecting our yeast to not only thrive in it, but also produce a lot of cellulases and also be quite happy to do fermentation whereby it produces ethanol, which is also toxic to the cell. So there's a lot of work that goes into this robustness of fermentation organisms. So as I mentioned, to enable cerevisiae to hydrolyze cellulose, we have to express these three different types of cellulase activities. It is one of the most central parts of improvement that we need for developing cerevisiae as a Cvp organism. So as it happens, I was involved in research to develop yeast for CvP. We synthetically produced genes, which means that they were made in codon optimized form for us to express in yeast, clone them onto episomal plasmids, and express them in yeast. And we selected some very strong candidates. We tested up to 20 gene candidates for each of the different types of cellulases. We got some very good gene candidates, and we combined them in a yeast strain where we expressed all three of the cellulase activities, which was done in collaboration with a company called Moscoma in the USA. And we created this yeast, which we called Moscoma 1.0. It was very good at fermenting amorphous celluloses. So we looked at different types of cellulose substrates, and the ones that weren't that tightly packed into a crystalline form, which you call an amorphous cellulose, those were actually quite easily fermented by this yeast. And our Cbp strain was able to, on various amorphous cellulose substrates, of which paper sludge is one that you can get from industry, although you can chemically produce them in the lab as well. Our yeast did quite well without the addition of any extra enzymes. To work on crystalline cellulose, though, we identified that we actually need much higher expression of cellulobiohydrolase. But even so, our yeast was able to attain the same amount of ethanol as the parental strain when we added a lot of cellulases to it. We were able to save about 60% of the cellulases required in a fermentation like that. So our strain did quite well, but required quite a lot of improvement, and the research after that is ongoing, although unfortunately I don't have collaboration with Moscoma anymore. I do have collaboration with this Japanese group from Kobe University, who are creating very similar strains to us in that they are also producing cellulases in the yeast Saccharomyces cerevisiae. What they do differently, though, is they allow these these enzymes to be anchored on the yeast cell surface. So they produce the cellulase as a fusion gene with a cell surface protein, and that allows the cellulase enzyme to actually get stuck to the cell surface. So the idea is that this cell comes into contact with cellulose, and your enzymes are right there on the cell surface. It'll degrade the cellulose, and the glucose can be taken up by the cell. Incidentally, they made a similar strain for xylan hydrolysis, xylan being a polymer of xylose, one of the predominant types of hemicelluloses. And they were also, in both cases, they were successful in showing the conversion of the polymeric substrate into ethanol, although at relatively low levels in both cases. So we're currently collaborating to ascertain if a combination strategy would improve yeast Cbp strains. Should all the enzymes be attached to the cell wall? Should all of them be secreted in the way that we do it? Or should some combination of attaching some of the enzymes to the cell wall be done with the secretion of the free enzymes? So the advantage of putting these enzymes on the cell surface is that as enzymes benefit the cell directly. So these enzymes break down the cellulose, and the released glucose is taken up by the yeast cell. Whereas if we are secreting the enzymes freely, those enzymes are in the fermentation medium, and any organism that is in that fermentation medium gets the benefit of the hydrolysis products of those enzymes. But the disadvantage of putting your enzymes on a cell surface means that some of these enzymes may not work as well. You'll remember that we said cellulobiohydrolases are processive enzymes. They need to attach to cellulose. They need to walk along the cellulose chain and release cellobios in the process and peel those cellulose chains off the polymer. And we're not sure how well these processive enzymes will do when they are attached to the cell wall. So this is something we are currently investigating. Cbp organism development for cellular lytic organisms. If you're starting with an organism that grows on cellulose already because it makes the enzymes, they often, when you engineer these, it's to try and get them to produce more of the product. So produce more ethanol or another desirable product. And this type of research normally focuses on bacteria because filamentous fungi that also grows well on cellulose grow very slowly, and then they produce little or no ethanol, and they generally have very poor product forming abilities. But there is great advantages of starting off with a cellulytic organism for CBP, in that you can leverage the natural synergies between cellulases. These organisms tend to make a whole scope of different types of cellulases. They also inherently regulate the expression of the genes that encode those cellulases. So as your substrate change, you get different ratios of enzymes being produced that is optimal for the degradation of your cellulose. And many cellulytic and fungal and bacterial strains also metabolize hemicelluloses and the pentoses in the substrate. So you can theoretically have an organism that you don't need to engineer to also produce or to also use your pentose sugars. One of the organisms that's very interesting in this background is Clostridium thermocellum, which is an anaerobic thermophilic bacteria that produce these cellulytic enzymes on a structure called a cellulosome, where you have different proteins that attach to each other and to the cell wall, and it's a very efficient way of breaking down cellulose. And there have been several attempts to produce methods for the Clostridium thermocellum manipulation. It's not an organism that is as easily genetically manipulated as, for example, E. coli is. It's quite a challenge, but there has been some substantial work into increasing the amount of ethanol that this organism produces. Clostridium cellulyticum is a related mesophilic organism that was engineered to produce isobutanol directly from cellulose in the first demonstration of Cbp for a next generation biofuel production. So one of the problems with the Clostridia is that they tend to produce naturally this mixed acid type of fermentation, which means that in addition to ethanol, they're also producing lactate and formate and acetate. And this means that your carbon from your cellulose or cellubios, as it's written here, will be diverted into these different areas, and you're not going to make much of your product that you want in industry, which is ethanol. So how do we get around that problem? We need to solve this by increasing our product yields, and we need to also, in this case, in using bacteria, increase the fermentation robustness of our organisms, because unlike with Saccharomyces cerevisiae, these organisms tend to be very sensitive to the fermentation inhibitors that you get after pre-treatment. But in order to improve the amount of the product of interest, what we need is metabolic engineering. So metabolic engineering is the practice of optimizing genetic and regulatory processes within cells to increase that cell's production of a very certain substance. So we looked at our mixed acid production that we had over here. We're getting all of these competing pathways that are allowing production of competing products that take away carbon from the ethanol that we want. So in case anybody who had second year biochemistry with me need to be reminded, metabolic pathways are a series of biochemical reactions that reactions and enzymes that allow cells to convert precursors into molecules necessary for a cell's survival. And in metabolic engineering, there is often mathematical models of these metabolic pathway networks that calculate the yield of a useful product and often allow us to pinpoint the part of the network that actually constrains production of that product. So recombinant techniques can then be used to modify these pathways or network of pathways to relieve these constraints and allow us to get a high level production of our product of interest. So the ultimate goal of metabolic engineering is to optimize the production of a valuable substance on industrial scale in a cost-effective manner. This is exactly the type of techniques that was used to allow the production of ethanol from Clostridium thermocellum by knocking out all of these competing pathways. There are different ways in which you can do metabolic engineering that we'll discuss up next. But we can also just be aware that changes can have a drastic effect on a cell viability. You can't simply knock out any pathway in a cell. It might be required for the production of a molecule that the cell has an absolute requirement for, which means the cell will die if you knock out a certain pathway. You've got to be a little bit more clever. There are trade-offs between the cell's abilities to produce the desired substance and then its actual survival needs. One example of that is when producing certain amount or certain fatty acids that are very important in industry. You cannot knock out the cell's ability to make its own sterols. These use the same precursors, but if the cell cannot make its own sterols, it cannot actually survive. And so what people have done is they've down-regulated the production of the cell's own sterols while still allowing it enough to actually survive, and in that way improve the product formation of the product that we want. So common metabolic engineering techniques are the overproduction of a rate-limiting enzyme in a pathway. So looking at these metabolic pathway networks, we've calculated that there's one area that slows down the flux through our metabolic pathway, and we're going to try and overproduce that enzyme. Blocking competing metabolic pathways, most often by knocking out certain genes that will mean that the cell can't make certain enzymes. Heterologous expression of a brand new pathway might be one option, or engineering a native enzyme to work better. So looking at a little metabolic pathway here, we'll see a substrate coming in and different products that can be produced. And there's our product of interest. So using metabolic engineering techniques, we can, for example, if we calculated that enzyme 7 here slow down the production of our product of interest, we can overproduce enzyme 7 by either a heterologous production or overproducing the native enzyme that allows this reaction to happen. Blocking competing metabolic pathways, as I said, will simply be by knocking out the enzymes that are used in those competing pathways. We could also use an intermediate product. We could be interested in an intermediate product that can be used in a brand new metabolic pathway that we're putting in through heterologous gene expression. That would be a different type of metabolic engineering. And of course, we can, for enzyme engineering, let's say, the enzyme that is used in this step is not working optimally, we can make different mutations of this type of enzyme and try and isolate one that actually works better. That would be an example of enzyme engineering. So as an example of how metabolic engineering has been applied, there was an interesting article a number of years ago with researchers who worked on Escherichia coli. Now, E. coli cannot utilize lignocellulosic polymers, and it cannot really produce a biofuel. But through metabolic engineering, this organism was engineered to hydrolyze xylan and also some cellulosic substrates and produce biofuels or biofuel precursors. Now, all of the detail here is not important, but I want to point out that the way they did this was they had a plasmid where they expressed some cellulase encoding genes to allow breakdown of certain cellulosic substrates. They had a plasmid which was encoding genes involved in xylan hydrolysis. They put these together into an organism, and they were able to get these organisms to grow either on a cellulosic substrate or a hemicellulosic substrate. They then introduced brand new pathways for the production of some of these biofuels or biofuel precursors, which were quite complex, quite a large number of genes that they introduced at the same time. The joy of working with E. coli is that you can do polycystronic expression, which means you can put a number of genes under control of one promoter, which is something that those of us who work with yeast and other eukaryotes are very jealous about. So in this way, they introduced new pathways for the utilization of lignocellulosic substrates in that case, and also to produce fatty acyl esters, butanol and pinene, which is a jet fuel precursor. And when the combined strains, these strains could produce the product from actual pre-treated woody material, pre-treated switchgrass. However, the production levels here are very low, almost ridiculously so in terms of milligrams per liter of these products that are formed. But it does prove the concept that you can actually engineer an organism that could neither use a certain substrate to use that substrate, and then also that could not produce a certain product to actually produce a range of products. Finally, for this lecture, we will talk about biorefineries very briefly. What is a biorefinery? It is a facility that integrates biomass conversion processes and equipment to produce fuels, power, heat, and value-added chemicals from biomass. So we've been talking about biofuels, and you might get the idea that we are going to take a feedstock and just turn it into a fuel. And if you look at it in that way, it's become clear that that is very rarely economically feasible. So what we want to do is we want to improve the product range that we produce, and in that way make the entire process more economically feasible. So this would be analogous to petroleum refineries, where you actually start off with a single product, crude oil, and then produce a large range of products, energy products in the form of diesel and petrol, but also synthetic rubbers and plastics and all kinds of chemicals that go into everything we use every day, from clothes to cosmetics. So the general concept of a biorefinery then is that you do a fractionation of your biomass. You get your carbohydrates out, which you might do different things with. You might ferment it, you might add it to food and feed ingredients, or produce bulk chemicals from it. And then other fractions such as lipid fraction or protein fraction, depending on what your biomass is, there'll be various amounts of these, can be turned into other higher value products of interest. So biorefining then is the sustainable processing of biomass into a spectrum of bio-based products. We're not using, or we're not producing just one product, we're producing a range of products. So these will include, or could include, food, feed, chemical materials, as well as energy. So we're producing higher value products as well as energy products in a biorefinery. Biorefineries take advantage of various components in the biomass and their intermediates and maximize the value derived from the biomass feedstock. We could produce several low volume, high value chemicals, as well as low value, high volume liquid fuels. So high volume, large amount of fuel can be produced in this process, but that is a product that we know sells at a relatively low price. But we can also produce some of these specialty chemicals in a biorefinery, which might sell at a high price. And the combination of those selling prices can make our entire process become economical. So why do we need that? In terms of sustainability, and we can't just really talk about biofuels production, because if you look at a barrel of oil, it is actually used for a large variety of products. It is used to make diesel and gasoline, but it's also used to make jet fuel, it's used to make asphalt, it is used to make a lot of other chemical products that we use in our day-to-day lives. And for a second generation technologies to be successful and for us to be moved to a more sustainable future, we actually have to start thinking about producing these other products in our biofuel production facilities as well. In other words, move away from biofuel production facility into proper biorefineries. Biorefineries also generate electricity and process heat, which is referred to in industry as combined heat and power, for its own use, for use at the facility, but can also be sold to local utilities. The higher value products will increase the overall profitability of the biorefinery. The high volume fuel products help meet the energy needs, which is why, of course, we want them in the first place to produce those biofuels. And the power production will help to lower energy costs and reduce greenhouse gas emissions in the overall process. So future biorefineries might play a major role in producing chemicals and materials that are traditionally produced from our petroleum. Okay, so what are the factors that are critical to the success of biorefineries? You need a good technology base. First of all, You need very high conversion efficiency. You need to catch up with petroleum refineries that are that have over 100 years of experience in perfectly fractionating crude oil and make a large amount of very high value products from it. So we need a fractionation technology that is mature, and these days, from most of the interesting agricultural waste feedstocks, we are nearing maturity in terms of the fractionation processes. We need an integrated use of different technologies to ensure the total use of raw materials. There can't be any waste in the process. The plant fractionation has to be optimal. The enzyme technology, fermentation technology needs to be optimal. We need optimal metabolic engineering of the organisms that we use to make the different types of chemicals that we want to produce as products. And what we can't use in the biological way, we need thermochemical conversion to produce our combined heat and power, and perhaps some other higher value chemicals or finished fuels. We need product diversity. As we said, it's important to have a broader product range to remain competitive. We need that low volume, high value product, which might be a number of different products, a number of different chemicals, and then our low value, high volume products such as energy, and we can adjust these with market demands. So what we mean by that is, if the price of the fossil fuels drops very low, and it means our biofuel that we're producing in our biorefinery is no longer viable to sell because it's just too expensive to produce, then we decrease the amount of fuel that we make and we increase the amount of the higher value chemicals that we produce at that stage. And producing a broad range of products helps you to adjust to these market demands. What is also quite often produced in these facilities are food and animal feeds, which can help you with some of these higher value options that you require. You need it to be a large-scale operation. You need to exploit economies of scale. It was with most biofuel productions. I did say in an early lecture that you can make biodiesel if you have waste oil at a relatively small scale. You can make biogas at a relatively small scale. But in terms of a biorefinery, you need to work at very large volumes in order to be economically feasible. And cluster formation and integration has proven to be very important. It's allowed industrial synergies where one type of company might have a very good fractionation process, and then they do the fractionation of the biofuels, and then they hand the different fractions off to different companies that are very good at a certain type of fermentation or a certain type of animal feed production, for example. And this also helps you with that problem that I mentioned earlier, which is intellectual property protection, where the different companies might be built around their own intellectual property, and alone they don't really have a full process. But by working together in a cluster such as this, they can form an integrated biorefinery. Right, this is the end of this lecture. In the next lecture, we will look in detail at some more metabolic engineering of the yeast Saccharomyces cerevisiae to allow to produce products that might be interesting to produce in biorefineries.